Go ahead and introduce now our speaker, Dr. Sven Bocklandt from Bionano. And Sven, I'll let you take it away.
Yeah, hello everyone. Thank you for the introduction. So you're right, my name is Sven Bocklandt and I'm at Bionano. I'm the senior application scientist here. I just want to warn you that I have a cold and that when I hit my mute button, AT&T Connect seems to disconnect. So if you hear me make odd noises, that's me trying to suppress my cough to not hurt you too much. All right, so Bionano, I assume that most of you have heard from us. We spun off from Princeton University 13 years ago, and we've been commercial for about 4 years now with an instrument called IRIS. The reason really why we exist is because genomes are just really difficult. This slide here shows you an example of a human genome, where only about 1/3 or so of the genome are unique sequences, and about 2/3 are repetitive sequences. And these repeats are very hard to assess with short-read sequencing alone, because of course if you get such 100, 150 base pairs from, let's say, a sign, how do you know exactly which one in the genome this read comes from? And so any changes that happen in these repetitive regions are often missed with next-generation sequencing. This is true in this case the human genome. This is sometimes even worse in animal genomes, where we can see up to 95% repeats. So this is really, really hard to make sense of with existing technologies. So Bionano is not a sequencing technology, and we do not provide you next-generation sequencing. We do not provide you actually single base pair resolution sequence. Instead, what we're doing is we're showing you long molecules in its entirety. So what we do is we linearize these long DNA molecules in nanochannels, and I'll explain to you in a second how that works in more detail. But what you're looking at right here are simply molecules of DNA. Now, I've been a molecular geneticist for over 20 years, and I've never actually seen DNA molecules until I saw this data from Bionano. And what you're looking at right here is raw data generated by our instrument. So every vertical line is a single molecule of DNA. The backbone is stained in blue with an intercalating dye. And then these green dots are specific sequences that we have labeled. You see on the left here that there's these evenly spaced labels. Those are simple repeats. On the right here you see patterns of more complex repeats. And these kind of difficult to assess parts of the genome are often called the dark matter, because just like in the universe, that dark matter is really invisible with our current technology. So we've been around for about 3 years with the IRIS system, which automates imaging of these long molecules in our chips. With the IRIS chip, kits to prepare the DNA, and then a compute solution to assemble the genome and analyze it. Now, just 2 weeks ago at the AGBT conference, we introduced the IRIS the Sapphire system. I've been saying IRIS for many years. The Sapphire system, which has about 10 times the throughput of the IRIS system, reduces the cost for large genomes 6-fold, and is just much faster and much easier to use. For the type of genomes that you are working with, really both instruments qualify, and that's why I just wanted to make sure that I'm showing you both. Fundamentally, nothing has changed about the technology, how we acquire the data, or how we analyze it. So let me tell you how it works. First of all, we start with very long DNA molecules, and this is really important because, of course, we cannot show you the long-range information of the genome if we don't have long molecules. We have protocols that work for a variety of animal tissues. It works quite well on insect larvae or pupae, and also on adult insects. It's a little harder, but we've gotten that to work repeatedly. So when you use our protocol, which is really based on embedding the cells into agarose, doing the DNA purification while the molecules are inside the agarose, and then when you digest the agarose away, you have these very long chromosome arm length, sometimes molecules that come out. So once you extracted the DNA, we label that DNA at specific sequence motifs. So the way we do this currently is we use a nicking endonuclease. which is just like a restriction enzyme, except for it makes a single-stranded nick at a specific recognition site. So that's typically a 7 base pair sequence that occurs somewhere between 8 and 18 times per 100 kb in a genome. These enzymes are available from NEB, and there's 5 of them that we use and validate on our system. We have software that can tell you exactly which enzyme makes the most sense based on some sequence that you have from the organism you're studying. So typically for a genome, we use just one of these enzymes to recognize a single 7 base pair sequence. So once that nick is made, we use a polymerase, a simple Taq polymerase, with fluorescently labeled nucleotides to incorporate a label at these recognition sites. And so once these fluorescent nucleotides are added, a simple ligation will simply heal these molecules and maintain their long length. That sample that is labeled is then loaded into our chip, either the iris chip or the sapphire chip. And our core technology is really these nanochannels. These nanochannels are about 40 nanometers in width, very uniform in size, and so only a single molecule of double-stranded DNA fits in there, and these molecules are uniformly stretched. And because they're uniformly stretched, that means that all the information that we need to get from them, which is the length, the start and the stop position, and the position of each of these labels, all that information we can get by simply taking an image. And that's why our technology is called optical mapping, or we call it next-generation mapping, which is really high-throughput automated optical mapping. This optical mapping is actually done by the instrument, so either the iris or the sapphire, and we're pulling these molecules through these chips over and over and over again and image them in between. And so this differentiates us from older optical mapping technologies like Obgen, where the DNA was immobilized on a slide, and you could just not get the throughput that we can get. Just to give you an idea, on this Sapphire system, we can get 480 gigabase pairs per flow cell, which is really hundreds of X coverage for even very large mammalian genomes, and of course a lot more for smaller genomes. Iris is about 10 times less, so that even there, for a typical insect genome, you should get sufficient data out of a single chip. So these molecules and labels are then detected in these images, and so our data pattern that we pull out of that is again just the position of that label on these long molecules. And then on every single genome, we do a complete de novo assembly. Our software does not know what organism we're working with. It's not reference guided. There's really just a pairwise alignment of these molecules. And so we make a consensus genome map by aligning these molecules for as long as they can go. And so these genome maps typically have an N50 of somewhere between half a megabase and 5 megabases, depending on which enzyme you use and exactly how well the DNA was prepared. And then there's 2 applications. The first application does not involve any sequence, and it is calling structural invariance. And I'm going to talk about that real quick, just give you a few examples because I think it's relevant for some of the work that you guys are doing. So in this case, if in green you'd see a reference and blue is the genome map on the organism that you just assembled, you can see that there's a deletion here relative to the reference. So again, this is not sequence-based at all. This is purely done on the patterns, the spacing, and the number of these labels in the genome maps. And then the other application is hybrid scaffolding. They're really done in quite separate pipelines. And in hybrid scaffolding, we use our genome maps to anchor and orient sequence contigs, and then we output an assembly that is significantly longer and more accurate. And I'll talk about that in a lot more detail further down. So one more thing about these chips. I already talked to you about the nanochannels, but this is really what they look like. So we make them out of silicon wafers in the same factories that computer chips are made, using very, very similar processes. And on the chip, you see this pillar feature here. And so the DNA is, using electrophoresis, is being pulled through that chip, will gently unwind in that pillar region and then go into the channels. I can show you what that looks like right here. So what you're looking at on the left here are Gaussian coils of DNA The DNA is going through these pillar regions into channels. And if you wait a little bit from here on, every green line that you see is a single molecule of double-stranded DNA that is being pulled through these nanochannels. And of course, this is in multicolor. This image only shows you one color. This is again starting it all over. I've said long molecules many times. We start the analysis with molecules that are 150 kb in length, and then we go all the way up to about 2.5 megabases. These are the longest molecules that we can image in our system, but 150 kb and above is really what we're working with. So it's orders of magnitude longer than what even long read sequencing gives you on average. So let's talk about structural variation calling, and I'll go over that just real quick. So here's a beautiful example of something that we can do. In green, you have the reference. In blue, you have an individual that we mapped, and you see that this 90 kb sequence is actually present in 4 tandem copies. Now, how can we know this for sure? Well, every horizontal line that you see below here is a single molecule of double-stranded DNA that was imaged in our system, and every dot that you see is a label that was detected. So on the consensus genome map here, every line again represents the consensus of the label underneath it. So in this consensus genome map here, we clearly see 4 copies of this element, but more importantly, we have molecules that span the entire 4 copies of that element. So that's 360 kb here. There's no way you can do this right now with long read sequencing, because even PacBio reads don't at all make it through all these 4 units. And so for longer repeat arrays, you really need to span the entire array in able to size it precisely. And again, these very long molecules can do that. So a main difference between us and other technologies is that structural variants are observed and not inferred as they are with sequencing, where you typically chop up the DNA in little bits, glue it back together using algorithms, and then you hope that this gluing back together was done correctly, and that you can compare that with a reference or with another sample. So we just see these SVs on large molecules, and that's really the power of our technology. So this is an example of duplication. Here's another repeat that we can see very well. So this is subtelomeric in yeast, where the reference ends, and we can go into these subtelomeric repeats and give you exact copy numbers of those repeats. So using our newest algorithms, which are being released next week, you can merge data collected with 2 different enzymes for structural variants, and we have 90% sensitivity to detect large insertions and deletions. And more specifically, that's 99% sensitivity for homozygous insertions and deletions, and 87 for heterozygous insertions and deletions. And when I say large, that means 1 kb and above. This is spectacularly better than sequencing-based algorithms. We've done some experiments on the identical samples and cell lines with our system and with PacBio, for example, and our sensitivity is about twice as high. Here's an example of a translocation. Again, these very long molecules provide the supporting data to call this translocation. We have a number of labels that align with 1 chromosome, a number of labels that align with another chromosome. And we have 98% sensitivity to detect translocations, whether they're balanced or unbalanced. This slide gives you an idea about what else we can do. You can see 2 very complex rearrangements. Here's a large deletion that is called by these repeats here. And then there's these inverted repeats here that cause the inversion. And so all of these large rearrangements are just very easily visible using our system. So other than the SVs that I mentioned before, we can also call inversions, copy number variants, we can size repeat arrays, and we can visualize complex rearrangements. And so I think the main application for your work is that you can do this between species and visualize how the genome has been rearranged during speciation or evolution, really. Or you can do it between individuals and identify variants that could be relevant for traits. And again, this is all sequence independent. You don't need reference quality or any sequence in order to do these visualizations of the genome. We have a number of white papers on our website that go into a lot more detail. Here's one on plant and animal genome structural variation. We just posted something on bioRxives that will be submitted for peer review, where we actually go through the hard numbers and our sensitivity, and we show you the examples of these samples that we compared between us and other technologies. And all that is available on our Bionano website in the libraries section. So now let's talk about the second application, which is hybrid scaffolding. So hybrid scaffolding overall can give you about an up to 100-fold improvement in contiguity, reduce the number of contigs, and also correct assembly errors. So let me tell you how this really works. So in the hybrid scaffold pipeline, you simply import your FASTA assembly, and then we will output a FASTA assembly. So it's sequence in, sequence out. And what happens in between is that we will do an in silico digest with the restriction enzyme that was used to label the Bionano map. And we will use the pattern of that 7 base pair sequence in the sequence assembly to align the sequence with our genome maps. And that way, we simply align these patterns between our maps and the sequence contigs, and we can use our genome maps to bridge these sequence contigs and the other way around. Now, this works really well because Bionano maps typically break at what we call fragile sites. And a fragile site occurs when 2 nick sites occur on opposite strands close to each other, and so that would create a double-stranded break. Sequence contigs typically end at repeats. Now, we read right through repeats, and those are not an issue for us. So our contigs our genome maps almost always end in different spots than sequence contigs, and that's why there's such great complementarity in building these hybrid scaffolds. And so our software will then output a new assembly that contains the sequence from the 2 contigs in this case, oriented and anchored correctly. We did this early on with a few insect genomes. Here, the spider mite. This again was done several years ago based on a Sanger assembly where we scaffolded the sequence, and we got a great improvement both in the number of contigs being reduced, and then the contiguity improved significantly from the sequence assembly. Another advantage, of course, of the scaffolding is that we can bring the right sequence fragments together. In this case, we created a silk gene, or not created it, but brought it back together by simply anchoring the correct sequence scaffolds together. There was a repeat sequence right here that caused that assembly to break. We can do this with PacBio sequence as well, of course, and that actually works really well because of the longer contigs that you typically get. Here's an example in the goats, and I want to use this to point out how our error correction works. So what you look at here on top is the genome map in blue, and then the PacBio contigs are below that, conveniently also in blue. And what you can see here is that this contig on the left aligns perfectly with our genome map. The right half of the bottom contig aligns with our genome map as well, but the left half doesn't, and so there's something going on here. And what our software will do automatically is look at this conflict site and verify the long molecule support that we have at this conflict site. And if we have many molecules, like here, that have a large number of labels on both sides of this conflict site, then we will have very high confidence in our assembly. And what our software will do is it will automatically cut the sequence contig and then re-scaffold. If we have poor confidence in our assembly, we will do it the other way around, and we will cut the Bionano genome map and then re-scaffold. And we see an incredible improvement in contiguity and accuracy. Our assemblies typically go about, the contiguity improves sometimes 2-fold over not having this automated conflict resolution. Now, of course, everything gets flagged, and so you can manually inspect these files and manually alter the decisions whether to cut that or not. And so here's something that comes out of a white paper that I'll show you in a second as well, so you can read about it in more details. But when we check the number of genomes and we check the automatic conflict resolution, we were able to confirm the overwhelming majority by simply inspecting both the genome maps and the sequence assembly. When we couldn't confirm that, that was typically because there was just not exact clarity in what happened there. So this automated conflict resolution works well and improves the assembly a lot. We can scaffold 10x libraries as well. This was the first published paper about this from Pui Kwok at UCSF, where you can see the original Illumina assembly on top, the 10x assembly below, the Bionano genome maps, and then the hybrid, which shows you a single scaffold here of tens of megabases in length. And I thought there was a summary slide that may have moved around. Here's another example of the hummingbird, where we did a de novo assembly and then scaffolded the PacBio. And what you're seeing here is that there's, again, a gene that was cut into two contigs that we were able to bring back together. There's a muscle kinase gene, which may just have something to do with the incredible flight performance of this animal. So you see the long molecules that were able to bridge these two sequence contigs. Here's another and an interesting example that shows you that we don't just improve the contiguity, but also the accuracy of the assembly. So if you look at this here on top, you see the PacBio contig showing this tandem repeat array. Now, if you look at the Bionano genome maps, you see that there's two separate haplotypes that we assembled, and both of them are actually different from the PacBio config. So what happens here is that the PacBio assembler collapsed this repeat, and we were able to identify the two haplotypes, and again, the two correct sizes. And you can see some molecule images actually here below that show you the copy number, and you can simply count the number of labels to get this copy number right. We did an experiment where we took, this is a human, actually this is Greg Venter's genome. We took a de novo PacBio assembly and downsampled it from 90x to 50x and 30x, and then scaffolded it with Bionano genome maps. There's a lot of data on this table, but I just want to point out the top and the bottom. So if you just look at the contiguity here, the N50 of 90x PacBio was 9.5 megabases. The contiguity of 30x is 11.5 megabases. So this is about $50,000 saved in sequencing by adding a Bionano genome map for a few $1,000s, and you get an assembly that's arguably just as good as a 90x PacBio. Of course, there's more to sequence accuracy than just contiguity, but this gives you an idea of how much you could really save by adding Bionano genome maps. So with this new software release that's coming out next week, we're also adding 2 enzyme hybrid scaffolding. And what this allows you to do is to use genome maps created with 2 separate enzymes to anchor and orient your sequence assembly. And it has a number of advantages. One is that shorter sequence contigs can be scaffolded. Now, one limitation of our data type is that we don't have a very high density of labels. Typically, there's several kilobases between each. So we need sequence contigs that are almost 100 kb typically to scaffold them. Now, using these 2 enzyme genome maps, we can actually scaffold fragments that are as short as about 50 kb. It depends a little bit on the genome, so these numbers are just indicative. But this 2 enzyme hybrid scaffold makes a great improvement. And here's an example on some Illumina only libraries and a PacBio library, where the input sequence, in this case, for example, 190 kilobase per N50, was improved almost 100-fold using the 2 enzyme hybrid scaffold. These were human assemblies. Here's some plant and animals as well, where you see similar improvement. And really, depending on the quality of the inputs, of course, the output is more or less spectacular. Oh, that's the slide that moved away. I apologize. So that's the 10x data again. There's a single enzyme, but this just shows you that the 10x library that was really quite good already got to an N50 of 33 megabases after scaffolding. So the last example I want to show you was done in a collaboration with Rockefeller University, and that's the mosquito that carries dengue, Zika. It's been in the news quite a bit and still is. And so here we used sequential hybrid scaffolding, I think, with 2 separate enzymes as well. And what we noticed when we scaffolded this assembly, that we got double the expected size. There's probably 4 haplotypes here because there were a large number of individuals that were used for the DNA prep, and so there was quite a bit of variation in there. And this is just an example of 2 haplotypes that we assembled where there's quite a bit of variation. So this is a slide from Leslie Vosshall just showing you that they tried a whole different number of assemblers on this data. For some reason, the dovetail scaffolding didn't work at all. Ours gave a significant improvement in the contiguity. And so, again, there's a lot of data here, but you're welcome to look at this later in more detail. Of the 2 assemblies, the Canu2 assembly was chosen. What I just want to point out here was that basically the quality of these assemblies was verified by checking how many conflicts there were between Bionano genome maps and the sequence assembly. And so this one clearly has the lowest number of errors and also one of the better contiguities. And again, you see a significant improvement from the original NGS assembly to the hybrid scaffold. This slide shows you how this was done. Sorry, there's a number of pop-ups. Oops. Apologies. This slide shows you are we still here? Yeah. Sorry about that. This slide compares how Dovetail and 10x performed. You see that here the contig size plotted versus the scaffold size, but the Bionano scaffolds were an order of magnitude larger, at least, than the other methods. The other methods were able to show you our scaffold shorter contigs as well, though, and that's again because of our resolution. So now, what about Hi-C? There was just a question that popped up about that. So Hi-C is able to scaffold these assemblies as well and get sometimes chromosome arm length contiguity. So I think, to be honest, if it's like a race for the best N50, we may not always stay on top. But there's really one big difference between Bionano and Hi-C, and that's really this. Hi-C really works by cross-linking the DNA in its native state, and that allows you to get entire chromosomes assembled. But I think there's going to be quite a few errors in that assembly, and Bionano works really in the opposite way, and it is linearizing the DNA, getting very accurate de novo assemblies, and especially by using these 2 genome maps, I think we can error correct these assemblies very well. Last thing I want to say really about this is that a number of human genome papers all really just use the Bionano, including this unpublished one with the Bionano 2 enzyme system, where we get a contiguity that's actually greater than the reference genome. We have a white paper about hybrid scaffolding as well that gives you a lot of numbers on the performance, and you can find that on our website. So to summarize, I would like to say that Bionano maps show you the true structure of the genome and are able to identify large structural variants and rearrangements with much higher sensitivity than using sequence-based methods alone. Combining NGS and Bionano Next-Generation Mapping produces assemblies of the highest quality, and Bionano hybrid scaffolding is agnostic to the sequence technology used. This is a point I can't stress enough. Bionano is really the only non-sequencing based scaffolding technology. So all the other scaffolding technologies scaffold NGS by doing more NGS. And so the risk, the chances that you're going to correct sequence-based errors that way are pretty small. Our technology is completely orthogonal, and so if you assemble a Bionano genome map de novo, and you assemble a sequence assembly de novo, and they align perfectly, well then you can be really quite sure that that is correct. If there's a conflict there, well then you have using the two-enzyme method, you have two completely independently built Bionano genomes on top of your sequence assembly, and that can allow you to correct these sequence errors, I think, with greater accuracy. At the Biogenome conference last week, Stephen Richards presented some examples of insect assemblies that were recently put together and collapsed to make them more contiguous. And I think errors that may occur with that kind of process could be very easily verified with technologies like ours. And I think for a lot of genomes, using our data really has become a standard. And I'd like to end with that, and I'm happy to take any questions you may have.
Great, thank you, Sven. I just want to remind everybody that if you have any questions, go ahead and send those to me, Anna Childers, in the participants list, or you can go ahead and email those to me as well. And we have received some questions already. I think you addressed the one about high-C, the comparisons with high-C. Another question here, for hybrid scaffolding, is the in silico NIC map made from exact matches to NIC sites, or can there be, for example, one mismatch to the site and that be used as well?
By default, we use the standard match. I think that we risk great inaccuracy if we're using variation in there. It's already only a 7 base per sequence, so I don't think you want to play around with that too much. So we don't, of course, need 100% match between the sequence and the genome map, and our algorithms are taking a lot of error models into account. So there's no problem with an assembly that is where you have questions about the base pair accuracy. Typically, we can still scaffold that.
Okay, great. Another question here. How much does contaminating DNA affect your results?
Contamination as in contamination with foreign DNA, I assume.
Yeah.
So that's a good question. So in reality, what happens is we build our genome maps by simply aligning these molecules that we detect. And so if there is DNA from multiple sources present, then we will assemble multiple genome maps from multiple species within the same assembly. And of course, since there's no reference guide whatsoever, we cannot distinguish that right away. Now, if you then were to scaffold a sequence assembly that is pure, then these contaminating genome maps will just not be used in the assembly because there'll be no matching sequences. If there are matching contigs present because the sequence assembly was contaminated the same way, then of course they will be scaffolded as well. Although typically we don't see them being integrated, so they would just be separate scaffolds that are made that way. Now keep in mind that we build consensus genome maps, and especially for hybrid scaffolds, we really look for that consensus. So if you have 10% of the molecules being present from another organism, then the majority of the genome maps will not actually be influenced by that.
Okay, and I think that might help also address the question, has Bionano been applied to metagenomics data sets, and if so, how well does it work?
Yeah, that's a great question. We get that quite a lot. So we've done an internal exercise, as you may, where we created a database of genome maps for a number of bacterial strains, and we then mapped a specific strain of E. coli and used these genome maps to pull out the exact correct match out of that database. So theoretically, it's possible. We've actually worked some with the FDA that had an interest in using this method to identify pathogens that were not easily identified using 16S sequencing. Now, overall, if 16S works, then that is going to be a more effective and more efficient method. But for those organisms that cannot be identified that way, in theory we could do this, but we ourselves have not done that. There's been one poster for a customer who did something similar that we've seen, but it's possible, but not a standard application.
Okay, great. Also, so I think you spoke a little bit about preparing DNA. Is it a specific kit that you're using? And so if so, what's the best kit for getting the high molecular weight DNA extraction in order to use the Bionano technology?
Yeah, so the basics of our technology are actually fairly common. If you follow our standard protocols, you buy agarose from here and an enzyme from there, and you use the chef gel kit from Bio-Rad. However, we have come out with an animal tissue kit that has a number of optimized buffers, and so I would highly recommend that you simply get our kit to prepare that. Now, we are working with a number of partners right now to evaluate automated sample prep. There's a number of players out there that claim that they can get high molecular weight DNA, and we are testing all of them quite literally that we know of right now to see if any of them is able to give us the quality DNA that is essential. So for now, I'd say just get our kits, which you can get by contacting us, and we hope to have automated solutions available in the future.
Great. I think you talked a little bit about this as well. Is there a big difference in performance within the heterochromatin versus the euchromatin aspect of the assembly?
So the truth is that in theory we are not affected much by repetitive sequences, and so we can handle both. If you're simply talking about the assembly, we see parts of the genome that go missing, and in overall we can assess the repetitive parts of the genome a lot better than any other technology out there, but there's still some parts that are of limits if they're really too dense, too repeat heavy over too long stretches of time. So for example, we can make it through most smaller centromeres quite well, but we fail at really long centromeres simply because we don't have molecules that can span the entire length, and therefore we cannot size them correctly. So the answer sort of is we can do euchromatin and heterochromatin just fine, but we cannot guarantee you that we get 100% coverage of the genome, which we don't, but we do cover a lot more than we typically see with sequence-only assemblies.
Great. Do you know if anyone has tested using PB Jelly or other programs in order to really fill in gaps after you have the hybrid assembly from Bionano?
Yes, we've done that, and some other people have. At the Biogenome conference last week, Eric Jarvis actually showed some examples of how we use PB Jelly with the high-C assemblies to get the longest contiguity, and he mentioned there that he had not done the same with the assembly scaffolded with Bionano as well. We've done that internally a little bit, and that works just fine. And again, the advantage of the Bionano maps is that even if there are gaps between the contigs within a scaffold, we size them correctly. And so even though we cannot give you the correct sequence connecting these two sequence contigs, we get the sizing right up to an accuracy of sometimes less than 100 base pairs. So that makes filling in these gaps with something like PB Jelly really realistic.
Okay. Next question. Have you mapped the the Bionano data to a phased assembly like those, and I think you did discuss this, from the 10x Genomics or the PacBio assemblies that are assembled using the Falcon unzip assembler? Do you have to have any special considerations with those phased assemblies?
Yeah, that's a good question as well. So in this mosquito example that I showed, this actually worked really well, and what we found was that there were a lot of errors in the PacBio phasing. I mean, there's errors in our phasing as well. In this one, particularly, it worked out quite well. In other genomes, sometimes it's been really difficult, and that is the case when there's really very little variation between the two alleles. So if there's very little structural variation, then we will not be able to to distinguish between the two alleles very well. And then in the hybrid scaffolding, sometimes these assemblies get collapsed even when you add a phased sequence assembly. We are reworking our hybrid scaffold pipeline to be able to do that much better, but that won't be done until probably the end of the year. Right now, like I said, it works really well with very heterogeneous genomes where there's very heterozygous genomes where there's really a lot of large variation between the two alleles. It doesn't work that well with those where there's less variation. And then again, none of these methods that phase are perfect, and so we see a lot of errors when we phase them.
Okay, and so that really speaks to the same sort of problems you're seeing then when you're dealing with a mixed population of arthropods due to small specimen size and that sort of thing.
Yep, that's right, and of course this works better with if you're able to inbreed them.
Okay. How fragmented can your contig assembly be and still achieve good scaffolding with the Bionano technology?
Yeah, so like I said, this new 2-enzyme hybrid scaffold is able to integrate about 50% more contigs into the hybrid than the single enzyme approach. And typically for a single enzyme, we were saying that contigs need to be about 100 kb in size in order to be scaffolded. Now that is down to something like 50 or 60 kb if you use the 2-enzyme approach. Now, like I said, this varies greatly between organism and organism, and it really depends on the density of the labels within that scaffold. And of course, with 2 enzymes, there's the 2 parameters of the density of each of the labels. In order for us to identify a contig, we need to see a specific number of labels within that contig. And so using 2 enzymes means we see twice as many, but also depending on the enzyme density within that contig, shorter or longer contigs can be scaffolded. So it's hard to give you exact numbers. I can tell you that the 2-enzyme method incorporates about 50% more than the single enzyme, and so we can really go to shorter contigs. But some pure Illumina only assemblies, within 50s of 10 kb or below, there's just not much we can do with that.
Wonderful. Okay, and I think we've got one more question, so if anybody has questions, please feel free to send those in. This question, is there a suggested recipe of technologies and approaches to use to get an optimal assembly when you're thinking about pairing perhaps short or long read assembly sequencing data along with the Bionano or other scaffolding approaches?
Yeah, I mean, that's really, the method that works the best seems to be the kitchen sink method where you use everything. And I think depending on your cost, the simplest solution that gives you the greatest accuracy seems to be PacBio de novo combined with Bionano. Eric Jarvis did a very thorough comparison of all the methods, and that is on the slides that show you accuracy is really the one that scores the highest. So PacBio and Bionano. If you want to, if you can't afford the PacBio for time or money reasons, 10x combined with Bionano works really well as well. I think for the greatest contiguity, adding high C helps. But then again, I think it's really essential, and I think we're going to see more and more evidence of that. That there's really some structural errors in a lot of high C assemblies from what we can see, and so adding us as a sort of corrective factor really would help there.
Okay, wonderful. I think that we've got all the questions answered that I've received so far. Is there anything else you'd like to add at this time?
We didn't talk about price much. So I want to give you just a quick idea of how expensive it is to incorporate it. So first of all, our software is freely available. If you want to play around with it, I would suggest you wait a week or two because we have a whole new release of web-based tools that are coming out that are a lot easier to work with than our current Windows-only iris view. So it's called Bionano Access, and that will be coming soon, and that is free. So if you have a Bionano genome map, you can just do the hybrid scaffolding and all that on your own for free without any extra cost. Getting a genome map, if you have the instrument in-house, depending on your genome size, starts at about $650 per genome, and that would imply that you use the iris system and you use a single flow cell. And again, for smaller genomes, that should be sufficient. If you use the 2-enzyme approach, then the cost will be double that, and so we're talking about $1,300, $1,200, $1,300. For larger genomes, Sapphire is better, and the prices are comparable there, but you get about 10 times more data. Now, if you do not have an instrument available, then of course you need to go through a service provider. There's a number of them, and we can help you find them through our website as well. But they're all over the United States and Europe and Asia. And for smaller genomes, typically, if they do the DNA prep, run the sample, do the assemblies, do the hybrid scaffolding for you, I think you can count on about $5,000 or so. But then again, that depends greatly from the size of the genome and what exactly you want and who's doing the work for you. And I think that's that's all I had.
That's wonderful. Thank you for giving us that cost breakdown of having the difference between doing at least part yourself or having it all done by a service provider. And I'm glad you guys have that list of service providers available through your website. I think that will do it then for today. I really appreciate you, Sven, for spending so much time with us and walking us through this technology. And I want to thank everybody who listened in and supplied questions. So with that, I guess we'll sign off and let everybody know that next month, Dr. Keith Hopper, who's one of the USDA scientists, a research entomologist, will be presenting, and we'll send out more information and reminders about that, both through the website and through the mailing list. So with that, thank you, everybody. I hope you have a wonderful day. Take care.
Bye-bye.